So this chapter is on prokaryotes, and so we'll be studying bacteria in this chapter. And in the first section, we're going to look a little bit about the basic diversity of the prokaryotes. And your first objective is to be able to differentiate between the archaea, the bacteria, and the eukarya. And then the second objective is describe the basic characteristics that we see in bacteria and eukarya, and then look at the classification systems for prokaryotes. There's a history of microbiology, and for the most part, most of human history, we didn't know about prokaryotes. They were too small to be seen with the naked eye. It is interesting that in 1546, an Italian physician by the name of Fracastoro suggested that diseases were caused by unseen organisms. So it wasn't really until new lines of technology were developed that we were able to visualize these microbes. Some of them do cause disease, and that's the microscope, so microscopy, and then the technologies used to study infectious diseases. Antony van Leeuwenhoek used simple microscopes that he worked on and refined to study microbial life, and so he accurately described some of these microbes. And nowadays we have these electron microscopes that allow us to see the ultrastructures of these microbes. So we can see their organelles and so on. And then Louis Pasteur, you might have heard of pasteurization. Louis Pasteur, they pasteurize milk, so they boil it and heat it to kill many of the microbes in there to prevent illnesses that you may get from consuming raw milk. But when it comes to Louis Pasteur, during those days, it was believed there was an idea of spontaneous generation, and he did some experiments that basically refuted this idea. And spontaneous generation means you can get life from no life, but essentially today now we know that in order to get new cells, you have to have the pre-existing cells already, and he did some experiments that disproved the spontaneous generation hypothesis. And then Robert Koch studied a disease called anthrax, and he came up with some rules to follow, or postulates, in providing evidence for whether or not a certain microorganism causes a disease. And of course, these things would be developed using laboratory animal models, not humans. So here, you would have to look at the microorganism, and that microorganism has to be present in those organisms that are displaying the disease, and then be absent in those that are healthy. And then you must be able to isolate that the microorganism that you think is causing the disease, and then grow it in a culture. And then you're going to take that culture in the third postulate, and then use it to infect a healthy host. When you do that, that healthy host must show the disease again, and then you must be able to isolate that microorganism again from the diseased host. So in the new host that you've infected, they're showing the disease, and then you're able to isolate and culture it again. And if you can do that, then you have strong evidence to show that that microorganism that you isolated is the one causing the disease. The prokaryotes are really quite diverse. They represent descendants of some of the oldest living organisms on the planet. They've been around. They were around and evolved 1 billion years before eukaryotes evolved. I remember thinking back to the prior chapter we had. The first prokaryotes are seen in fossils that are 3.5 billion years old, and then you don't see eukaryotes until 2.5 billion years ago. Most prokaryotes have not been identified. It's estimated that 90 to 99% have not been described or cultured. In order to describe them, you have to be able to grow them in the lab to adequately describe them. And it seems that there's a lot of evidence for them occurring in the soil. We talked about this earlier in the virus chapter of metagenomics. When you sample some soil and you get DNA that's bacteria-like, but then it doesn't match anything that they've grown in the laboratory, and that would suggest you have some new species of bacteria that they just haven't been able to grow. So based on that information, it's estimated that most of the bacteria have never been described. And then we have the two main domains of prokaryotes. There's the domain bacteria and then the domain eukarya. And for many of the eukarya, they are extremophiles, which means they love, philos means to love, extreme locations. In other words, they grow in some of these places where most living things could not. For example, in this hot spring in Yellowstone National Park, there's bacteria there that are capable of handling really high temperatures that most other bacteria or other organisms couldn't handle, and they refer to those as thermophilic. There's acidophilic, so those that can grow in well in acidic environments. When it comes to characteristics of prokaryotes, they're unicellular. Some may grow in associations, maybe even communicating, very simple chemical communication with each other, or in biofilms. Like the biofilm you see here in this electron micrograph, those blue structures there are millions of bacteria on that yellow structure, which is a bristle of a toothbrush. So this is bacteria taken from somebody's teeth. The cell size, they do vary tremendously, but they're small. Many are on the order of micrometers in size, and generally speaking, they're about 10 times smaller than a typical eukaryote cell. They don't have a nucleus, but they have a nucleoid region where they'll have one single circular chromosome, and it's made of double-stranded DNA. And they may have other small pieces of DNA that are also circular, and those are called plasmids. And those plasmids themselves may have codes for certain other proteins, basically contain other genes. Plasmids are typically picking up through horizontal transfers either from the environment or from another bacteria. We'll go over that in the next sections. And for by cell division, there's no mitosis here. There's no nucleus to divide. Instead, you start off with one bacteria. The bacteria grows, then it replicates its DNA, and then divides into two equal-sized bacteria, forming a new cell wall in between. That's referred to as binary fission. So never ever describe bacteria reproduction as involving mitosis. That's division of the nucleus. When it comes to genetic recombination, bacteria can display genetic recombination. Mostly it's through horizontal gene transfer, which I mentioned just a while ago. And there they would pick up some DNA from either another bacteria or from the environment or even from infection from a virus. So this is not a form of reproduction. The organism is not some new offspring from prior, and you're getting genetic recombination like you do in sexual reproduction. So this is not a form of sexual reproduction. This is just different, and it does provide some new variation of within the population of bacteria. There is no internal compartmentalization, and the reason for that is there's no membrane-bound organelles. And because of that, you can't separate metabolism like you can in a eukaryote. There's no endoplasmic reticulum. There's no Golgi apparatus. The plasma membrane, though, may have some extensive infolding. And what that's going to do is going to provide some surface area for some cell metabolisms like respiration and photosynthesis and photosynthetic bacteria. They do have flagella. Some bacteria do. It's simpler in structure and in the way it works. So it's very different from a eukaryotic flagella. The eukaryotic flagella is much more complex, and you might have covered that in some detail in your Biology 1 course, the eukaryote flagella. Some bacteria do have pili or pilus, which translates to hairs. These are little fine extension-like structures. They can be found on some. Sometimes they're referred to as fimbra or fimbri. And they have various functions. One may be for genetic exchange. I mentioned plasmids earlier. One bacteria may transfer a plasmid or a small piece of DNA to another one with the pilus. The pili can also be used to attach to surfaces. For example, many pathogenic bacteria that cause disease may attach to the surface of tissues with their pili. And then in some bacteria, the pili are used to assist in motility or moving around the surfaces of the structures where they grow. There is a great deal of metabolic diversity there. When it comes to photosynthesis, there's oxygenic, which means to produce oxygen by splitting water, just like plants do. And then there's anoxygenic photosynthesis. For example, in some sulfur bacteria, instead of producing oxygen, they produce sulfur. And then there's chemolithotrophic. And here these bacteria get their energy from electrons from inorganic compounds. So they basically will oxidize inorganic compounds, extracting those electrons as a way of them getting energy to build organic compounds. So bacteria and archaea do differ, and they differ in 4 key areas. They differ in their plasma membrane structure, the cell wall structure, the way DNA replication occurs, and in their gene expression. When it comes to the plasma membrane for the bacteria tend to have their plasma membrane is much more similar to that of a eukaryote in terms of the basic components. You start off with a glycerol molecule. A glycerol is a 3-carbon polyalcohol, and an alcohol has a hydroxyl group. So there would have been a hydroxyl group here, but through dehydration synthesis, you can add this fatty acid here. And the fatty acid has this functional group called a carboxyl group. And when you get that bonding there between your glycerol carbon, one of the 3 carbons there, and the carbon of the what was the carboxyl group, then you have your hydrocarbon tail here. That's going to be called an ester bond. Okay, this is different in archaea. In archaea, instead they have an ether bond. An ether bond is a carbon, oxygen, carbon. There is no double bonded oxygen out here overall. And then the hydrocarbon tails here are branched, and you can see the branching here, those little sticks coming out there. Everywhere there's a bend, there's a carbon and enough hydrogens to fill the octet when it comes to the basic chemistry of carbon there. So that's a big difference. That's one big difference there. And the other one is that in the archaea, they can form these tetraethers. So we mentioned what an ether is. That's the bonding between the hydrocarbon and the glycerol molecule. But they can form tetraethers, and that ends up making a monolayer instead of a bilayer. And here the one side of the molecule where you have your glycerol molecule is the hydrocarbon tail is actually bonded by another ether to the other side of the molecule. And so that makes one layer connected to the other one, making one actual layer, because the 2 sides of the membrane are covalently bonded to each other all along there. And that makes a monolayer instead of a bilayer. And the result of this, when you have this monolayer, because of the tetraethers along the membrane, is it provides more stability, especially for these, not bacteria, but these archaea that tend to live in extreme environments like really high temperature. A lot more energy when it's higher temperature, and that provides more stability when there's a lot more thermal energy in the system that the archaea live in. When it comes to the cell wall, all prokaryotes, archaea, bacteria have cell walls, but there's a difference. The bacteria, those in the domain bacteria, have their cell wall made of peptidoglycan. Remember, plants is made of cellulose, which is a complex carbohydrate. A peptidoglycan is a complex carbohydrate. Glyco means sugar, just like carbohydrates, but it's got some amino acids in there, some short chains of amino acids, so that's where it gets its name peptidoglycan. And that's typical of all bacteria, but for the archaea, they lack peptidoglycan. There's one kind of cell wall that's very similar called cetomurine. And it's similar to peptidoglycan, but there's other types as well of complex carbohydrate protein type membranes found in the archaea. When it comes to DNA replication, remember that all prokaryotes have a circular chromosome. Unlike eukaryotes have several chromosomes and they're linear, right? And in a eukaryotic chromosome, when you come to DNA replication, you're going to have several points where your replication is going to occur, so that you have several replication bubbles going all at once within one chromosome. But for a prokaryote, there's only one origin where replication occurs. And in that origin, the replication bubble starts and then goes in both directions. So you're going to have a set of enzymes that include DNA helicase and DNA polymerase going in both directions until they reach the other side, and the end result would be 2 chromosomes that are exactly the same. So this is That's the same for the bacteria and the archaea. Where they're different are the details of the enzymes involved in this replication. And when you look at the archaeal DNA replication, the enzymes involved and the very specific details, the enzymology of how that occurs is more similar to that of eukaryotes. And this gives us a clue on which group of prokaryotes is more closely related to the eukaryotes, and that would be the archaea. And then the same with gene expression. So not only when we replicate DNA using DNA chromosome to make a replicate of itself, exact copy, gene expression is when you take your DNA, you take a segment of DNA, which contains a code for a protein, and then you transcribe it to make your RNA, your messenger RNA, and then translate it to make your protein. Making that protein depends on the code within that segment of DNA. That process from here to here, that's transcription, something we reviewed in the last chapter, and then translation occurs there. Again, when it comes to the details of this gene expression, the processes that have to occur for the archaea are much more similar to those of eukaryotes than for the bacteria. And so again, this ties. If we're going to use our new understanding of molecular biology, and there's a connection between your molecular biology and your phylogeny, your evolutionary history, this would suggest that the archaea are more closely related to the bacteria. So this brings us to classification. Prokaryotes are not easy to classify, okay? Especially if we're just going to go based on forms. When you do that, you're not necessarily going to end up resulting in a good representation of the evolutionary history of the bacteria or the evolution of the bacteria. So prior systems of characterizing were, again, based on these just simple observable phenotypes, like whether or not the bacteria was photosynthetic or not photosynthetic, whether they were motile or not motile. If they're unicellular, colony forming, or growing filaments. Filaments would be where when the cells divide, they just stay stuck together. So you might have a bunch of rod-shaped bacteria growing in a filament like that. And then whether or not they form spores or not, and whether the division is by a binary fission called transverse binary fission. Again, these are all things that are easily to observe. And then another way we might classify them would be on whether or not they're important human pathogens causing disease. Pathos means disease, and gen for generating. Nowadays, the molecular understanding is used for this classification. So looking at sequences in proteins, the amino acid sequence is determined by sequences in genes. So there is a relationship. The more similar the sequence of amino acids are in key proteins, the more closely related, that would infer a closer relationship between two different species of bacteria. The percent of guanine and cytosine, remember guanine and cytosine are the bases you find in DNA. So you look at the percent of guanine cytosine compared to your adenine and thymine in DNA, and that's another a bit of evidence for helping to classify the amount of hybridization that can occur. If you take DNA, a chromosome from two different species of bacteria, and then you split apart the chromosome, and so you only get one strand of the DNA from each one, and then you try to put the two from the two different species together, one strand from one species, one from another. How closely do they match each other when they start to form back to a double strand? The codes are going to be different, but the more similar the codes are, the greater that hybridization would be, and then that would infer a closer relationship. And then looking at RNA sequences, the ribonucleic acid, specifically a ribosomal RNA, the RNA that makes up the ribosomes themselves, that tends to be more stable over time, the RNA sequences within the ribosome, and so that would be good at trying to determine relationships. And then looking at the entire genome, the entire chromosome, and sequence that and then compare. So these are ways for classifying. And like I said already, even before just the similarities in the way DNA is replicated and in gene expression, going from a gene to a protein, suggests that the domain archaea is more closely related to eukaryotes. So we can see they're more close relationship there than the archaea are to the bacterial domain overall. And so then you go all the way back over here to some of the first cells that evolved. So the current molecular classification based on molecular data has been proposed, and there's a authoritative reference for bacteriologists. It's called Berge's Manual for Systematic Bacteriology, and there it suggests these specific groupings of bacteria. Keep in mind that there's still more to learn, and these, as understanding improves, things are likely to change here. And keep in mind also that most bacteria have not been described either. So this is a simple classification, relatively simple. It looks complex to us. We don't study this every day. Look over it once. I'm not going to have anything having memorized any of this, but you can see the familiar branching diagram here. It looks like a cladogram here. Over here you would have some common ancestor. To the left is where you have your archaea, and then to the right is where you have your bacteria. You have a group called the thermophiles here. These are gram-positive bacteria. We'll talk about some differences in gram-positive and gram-negative bacteria. These include organisms that do cause disease, including Clostridium, which can cause tetanus, which is where you cannot relax your muscles. And there's a tetanus shot for that, vaccine. There's another Clostridium species that causes paralysis and food poisoning. Then you have over here spirochetes on the southern branch here, and then the photosynthetic groups, including cyanobacteria, which we're going to talk about a little bit in the lab. And then another group called chlorobi, and both are photosynthetic. And then we have the Prokaryotic over here. They include beta and gamma. And gamma include enterobacteria. And entero, or enteric bacteria. Entero means your intestines. So this is within your digestive system. And anytime you say entero, that's something to deal with your digestive system. So these would be found in your gut, as part of your gut flora. And some of them are just more normal there. Others may cause disease, depends on the strain of bacteria. And then this other group out here, delta. Again, look over this, but don't labor any on trying to memorize anything in there. So in this section, we're going to look at prokaryote cell structure. And your learning objectives, first one is to describe the general features common to all prokaryote cells. The second objective is to explain why bacteria can stain gram-positive or gram-negative. And then number 3 for your objective is to describe the features that might discriminate one prokaryote from another. First thing we're going to look at in this section are some of the shapes that bacteria can come in. There are 3 basic shapes, and the cell wall helps maintain the shape. The first shape is rod-shaped, and that's technically called bacillus, and plural would be bacilli. And then there's coccus, which is spherical shaped, like you see in the second image there, and cocci would be plural. And then spirillum, and spirilla would be plural, and these would be twisted shape. Spirilla would be the plural form. So you see all 3 there on the bottom. Here's the rod shape. This looks like one is going through a binary fission right there. And then here's your spiral shaped bacteria. For the cell wall, for bacteria, it's made of peptidoglycan, which is modified sugar, complex carbohydrate with short peptide chains. I took an image off the web to show this. The individual sugars are modified with nitrogen-containing groups, and so these are your sugar rings here, and then you can see the amino acids that are bonded there, and then you have this cross-link with 5-glycine, so pentaglycine. So you would have these sheets of this material, which is peptidoglycan. Now, the purpose of the cell wall is to provide protection that helps maintain shape. If you're in a hypotonic environment, that means the concentration of water is higher outside, and so water would tend to come into the bacterial cell. And so this the cell wall helps keep the cell from lysing or bursting. The archaea does not have peptidoglycan that was mentioned earlier. There is a similar compound called pseudomurine in some archaea, but there is a greater diversity. So their cell walls are made of other kinds of polysaccharides with protein in there. So that's something to remember for bacterial peptidoglycan. When it comes to the bacteria, they can stain either gram-positive or gram-negative, and this is for the bacteria. And so this can be useful in characterizing your bacteria. The gram-positive bacteria in the gram stain are going to have a thicker cell wall, peptidoglycan, and they'll stain purple. So that's something to remember there. So gram-positive, purple, and the reason for that is the thicker cell wall. For gram-negative bacteria, it's a thinner cell wall, so there's less peptidoglycan, and so they won't retain this purple color. Instead, they're going to counter stain with another stain that's pink. So there's two different stains that are used during the gram staining. And this is the gram stain. Overall, you get your sample of bacteria and you would put it you would fix your bacteria onto a slide, a microscope slide, and then you put different stains on there. In fact, in the laboratory, there's supposed to be consideration of the gram stain now. Because this is online, we're not necessarily going to be doing the gram stain, but that doesn't mean that we can't see video of the process. And I think I even found a simulation online that you can work through to help you better understand the gram stain. You are responsible for understanding how it works and why it works. So after you would heat fix your sample of bacteria on there, you basically going to treat the bacteria with one stain called crystal violet. So that would be your initial staining. And at this point here, if you're gram-positive or gram-negative, both are going to take in the dye and look purple. And then once you do that, then you're going to rinse. You would rinse with water, gently rinse with water. And then you're going to put iodine on there. Now, iodine itself can serve as a stain, but the iodine, what that's going to do is the iodine is going to form a complex with the crystal violet. And this is within the cell wall. You remember that on the earlier slide, there's a thicker cell wall in a gram-positive bacteria. But at this point, both the gram-positive and the gram-negative, and they're supposed to be represented here. This would be gram-positive. This would be gram-negative. They both still look purple at this point. And then you're going to decolorize with an alcohol. So if you run, wash with, say, an ethanol, that's going to clear out and decolorize any of the stain that's on there. And unless you're a thick cell wall, it's harder to rinse it out. And the alcohol ends up dehydrating the peptidoglycan, and that traps the gram stain crystal violet within there. And keeps it purple, whereas the gram-negative is going to decolorize and won't have any color. And then after that, you're going to do a counter stain, and the counter stain is safranin, and it's a reddish color. And so when you put the safranin on there, both bacteria types are going to take the stain on. But because the gram-positive retain that crystal violet, it's going to maintain and continue to look a dark purple color, whereas the gram-negative are going to look red in color. So here you have an image on the bottom that shows two different types on the same slide, and so you can see some gram-positive and then some purple. The pink is the gram-negative, and then the purple is the gram-positive, right? So this would be gram-positive, and this one would be gram-negative stained. And so there, you have two different types of bacteria. We don't know exactly what they are, but we know one's gram-positive, one's gram-negative. So looking at the gram-positive, the cell wall is actually thicker. And the image here on the side here shows that thicker cell wall. Cell walls are always on the outside. Here's your plasma membrane right here. And that's supposed to represent your peptidoglycan wall. It also contains other compounds, including lipoteichoic acid and teichoic acid. You can see those in there embedded within the membrane, giving the cell wall, not embedded in the membrane, but embedded in the cell wall, giving the cell wall certain properties. If you're gram-negative, the cell wall is thinner. So you can see that much thinner peptidoglycan wall there. Here's your plasma membrane on the inside. And then they're going to have a second membrane on the outside that has phospholipids on there. It also has lipopolysaccharides, which are these structures here, and then some proteins called porins that are there. And so you have a second membrane on the outside for gram-negative. But the key here for the gram stain is that thinner peptidoglycan wall that doesn't trap the crystal violet. And so the gram negatives also show some resistance to many types of antibiotics. So when it comes to treating, let's say you have a bacterial infection of bacteria that can cause disease on there, it helps to know what kind of a bacteria you have. Is it gram positive or gram negative? That's going to determine what kind of antibiotics you're going to use. And then some bacteria have another layer, a rigid layer called an S layer. That's not imaged here, but this would be another layer. It's a rigid, sort of crystalline-like layer found in some bacteria and some archaea. So that's your S layer here. And this is going to be outside of the peptidoglycan or outer membrane layers in gram-negative and gram-positive bacteria. The functions for the S layer are diverse and often involve sticking to surfaces or adhesion. And then some bacteria are going to have a capsule, and the capsule is a gelatinous layer found in some bacteria. That also aids in attachment and can help protect the bacteria from being recognized by the immune system. And this one here, this image that I found here to share with you, shows a bacteria. It shows a flagella there. It even has the pili there. There's a pilus, a singular layer there. And here, this one has the capsule on the outside. So that would be your gelatinous layer there. And then here's your cell wall. So it's made of peptidoglycan, and then the plasma membrane on the inside. There is no nucleus. And while the chromosome is going to be circular, it can be relatively large. So it's going to be coiled up, but there's no free ends to this, not a linear chromosome. The bacteria have, some bacteria have flagella as well, and the flagella is simpler in structure. It's made of a helical protein called flagellin there, and then there's a hook-like structure to the bacteria. The eukaryotic flagella is more complex, and it is made of microtubules, and there's a specific arrangement in there. This one was covered back in Biology 1 chapter in there. You can see that this one here is, this one would be a gram-negative bacteria. You can tell because it has this outer membrane here, and then there's the cell wall there, and then here's your plasma membrane here. But overall, the structure here at the base is going to have these structures here on the bottom that look like ring-like structures here. And this structure overall, you can see in the electron micrograph image over here, you can see those outer and inner ring structures here. But overall, this is a motor here. So the bacteria maintains a gradient of protons, or positive protons are basically hydrogen ions without their electron. And if you have a higher concentration of protons out here, just outside the membrane, when we allow them to come in, that creates a spinning motion at the base of this flagellum, spinning the hook, and then that causes the flagella to whip around like a propeller. And this would pull can move the bacteria through the medium that it's in, so involved in locomotion. The motor structure here is very, very similar to the APT synthase that you see in mitochondria in eukaryotes. Remember, the mitochondria is originated as a free-living bacteria outside. At least that's the evidence that strongly suggests that. And so perhaps there is the motor structure there may be homologous to the APT synthase. As protons move through there, that causes a spinning motion in that enzyme, which is used to make ATP. But that's the mechanism here for flagella. It's spun around with this motor. And then we have pili. Pilus is singular, and these are the hair-like structures that have already been mentioned, found in gram-negative bacteria. And as mentioned before, they can aid in attachment to surfaces, and whenever bacteria are exchanging plasmids, they form this conjugation tube that's a modification on the pilus structure there. So one bacteria will form a pilus that connects to the other one, and then can transfer a plasmid to the other one. Some bacteria form endospores, and these are thickening of the wall. What happens when there's a lot of stress in the environment, maybe there's not enough water or nutrients available in the environment, the bacteria will lose a lot of its cytoplasm, keeping only some major components in there. Of course, its nucleoid region, where its genome is located, and form this very strong resistant coating around it. These are called endospores, and they can resist a great deal of stress in the environment, including heat. And so when conditions become right, the endospore germinates. Its metabolism becomes activated again, and then begins to grow and divide and so on. Some bacteria that do cause disease in humans, like tetanus or botulism and anthrax, form these endospores. Tetanus is found in soil, and if it gets in to a deep wound, when you get cut, that bacteria goes in there and then begins to germinate and then produce a toxin that causes tetany, and tetany is when your muscles cannot relax, and you can die from that. Botulism is sort of the opposite. It causes a paralysis because of the toxins produced. In fact, they use that same toxin for Botox therapies, where they to reduce wrinkles. Kind of weird. Prokaryotes also have complex internal membranes. They don't have membrane-bound organelles, but they do have enfoldings here. You can see the enfoldings in this bacterium here, this electron micrograph. Those foldings there provide surfaces for things like cell respiration. So this would be an aerobic bacterium, and the process of cellular respiration where ATP is made. So this might be similar to what we look at when we were looking at a mitochondrion. They had this folded inner membrane, and those foldings are called crista in there. And so those membranes provide surfaces where ATP can be produced. And then here's a photosynthetic bacteria, and then you can see these folded-like structures that are within there. And those folded-like structures are similar to thylakoids found in chloroplasts. And remember, chloroplasts also have a lot of evidence to suggest that they were once free-living photosynthetic bacteria as well. Now, these are not membrane-bound organelles. They're not part of the endomembrane system found in eukaryotes. So this is just an elaborate folding of the plasma membrane inward to provide surface for these metabolic processes. Remember that prokaryotes, they don't have a nucleus. They have a nucleoid region. And they have a single circular chromosome. And they may have other bits of DNA that are also circular, called plasmids as well. Their ribosomes are smaller than eukaryotes, and this is one of those bits of evidence when you look at a chloroplast or a mitochondria. They reproduce by binary fission just like bacteria do. They have their own ribosomes, and their ribosomes are smaller than the ribosomes found outside in the eukaryote. But so we see that similarity. So in prokaryotes, these ribosomes are smaller as well. And so they differ in their RNA and protein content. So a ribosome is a combination of ribosomal RNA and protein as well. And then that one difference allows for a target for antibiotics. So the antibiotics can affect translation, right? Because if the antibiotic can affect the ribosome there, then you can't make the protein, and that affects translation. And that can stop the metabolism and help end that bacteria. And if these antibiotics are specific for that kind of ribosome, then it wouldn't affect the ribosomes of the rest of the cell of the eukaryote. So there's potential there for those types of antibiotics to work. So in this section, we're going to look at prokaryotic genetics, and your learning outcomes are going to be to compare the mechanisms of DNA exchange in prokaryotes, and then your second objective is to describe how genes are mapped in Escherichia coli, which is bacteria found in your large intestine, and then describe antibiotic resistance and how it can spread between bacterial populations. So that's your third objective. And we're going to start by recognizing that bacteria do not reproduce sexually. There's no sexual reproduction in there. And so that's not a way to get genetic diversity in a population. We know sexual reproduction combines chromosomes, genetic information from two different parents, and then recombines them and reshuffles those genes so that you get different combinations from the two parents. That doesn't happen with bacteria, but there are ways that bacteria can increase their diversity within their populations. And one of those ways is by a horizontal gene transfer. And there's three types of horizontal gene transfer. And that's just, instead of transferring genes on to the next generation, that would be vertically. Here, the bacteria cell is already alive and instead takes in genetic information from the environment in some way. And that new genetic information can lead to changes genetically, changing overall the nature of that bacterium or the phenotype of it, and that leads to variation. Now, when that cell divides, now you can have a new strain of bacteria. So there's three ways in which horizontal transfer can occur. The first one is conjugation, and this involves two different bacteria cells combining together. And then there's transduction, and this one here, a virus is going to transfer a piece of DNA to a bacteria. And then there's transformation, and here the bacteria just picks up a piece of DNA from the environment. It's important to note that all three of those processes have been observed in archaea. However, we're going to just focus on the bacteria in this section here. The first one we're going to look at is transformation, and there's natural transformation here. And this was actually observed, perhaps not knowing what it was called, in an experiments done by Frederick Griffith as the experimenter. He was working with Streptococcus using rodents, and there was two strains of Streptococcus pneumoniae is the name of the species there. There's the specific epithet. There was a virulent strain that had a capsule, which is colored in blue, and then non-virulent strain that did not have the capsule. And what he did was he had two groups of mice in his experiment, and he injected each group with the type of the Streptococcus, and the ones that were injected with the one that had the polysaccharide died. That's why virulence is bad. And the ones that got the non-virulent strain survived. So then he did another experiment where he heated the bacteria with a flame. You can see the burner there underneath the test tube there. Killed the bacteria, as you can see, is dead now, and so he colored it red inside to indicate it's dead, and injected it into the mice, and the mice didn't die. And then what he did was he mixed, so he got the two test tubes of living non-virulent, the non-virulent kind, and the heat killed one, and mixed them together in a test tube. Mix them together, and we know that the non-virulent doesn't kill the mouse, and the virulent, when you kill it with heat, doesn't kill the mouse. So you would expect that the mice wouldn't die. But when he injected with a mixture of dead virulent strain and live non-virulent strain, the mice ended up dying. And so the only thing that Griffith could conclude, not knowing about transformation, was that something must have been transferred from the once living deadly strain of the Streptococcus, and somehow got transferred into the living one, and then changed the nature of the non-virulent strain. We know that process today is now called transformation. And here the bacteria are actually able to do this because they're genetically programmed to do so. In other words, there are genes that allow for proteins that are important in taking in this environmental DNA from the environment. So this is just not accidentally picking up DNA from the environment. These bacteria have the genes that allow them to pick up this from the environment. So this is the actual image from this chapter here. If you have some bacteria that died, like right here, and then it lyses open, spilling out bits and pieces of its DNA, that DNA can be taken in by natural transformation. Now this segment of DNA that we see right here might match the same types of genes you see on a chromosome there. So this section right here would be homologous to this section here. And that's going to be the place where crossing over occurs, and we switch out. So this portion of the old chromosome gets removed, and then we insert the plasmid in there. And let's say that while you have the same kinds of genes there, it's possible to have different versions of the gene. Remember from classic genetics that was covered in your Biology 1 course, you can have different versions of the same gene. They arise by mutation. Mutations over time. Those are used. If we look at a flower, for example, you could have a species of flowering plant that produces white flowers or red flowers, right? Those are just different versions of the same gene. So what ends up happening now is that if that plasmid now that gets integrated into your chromosome, it may be carrying a different versions of the same gene, that now this bacteria is now going to be phenotypically different. And then every time it divides, it's produced a new population of bacteria that are genetically different than what the bacteria used to be. So this is genetic recombination that promotes genetic variation. Then there's artificial transformation. Some bacteria just do not have the genes that allow them to pick up DNA from the environment. In this case, you can force that transformation in the under laboratory setting. An example is for E. coli. So you can force genes into E. coli, Escherichia coli, and then they genetically engineer them to produce something like insulin. Insulin is a hormone that is produced by our pancreas, and if you have the type of diabetes where you no longer produce that insulin, you can get the bacteria to make the insulin for you and then use it to treat diabetes. And then there's transduction, and transduction involves bacteriophage here. And then there's two kinds. There's generalized transduction and then specialized transduction. Here we have to rely on our knowledge from the prior chapter on bacteriophages and the lytic cycle and the lysogenic cycle. Now, generalized transduction happens, it's an accident that occurs, and it happens during the lytic cycle. So during the lytic cycle, recall that your bacteriophage injects this DNA into the host cell and then hijacks the host cell's machinery and begins making copies of new viruses. When the viruses break out, the bacteria dies in the process. Okay? What wasn't really mentioned during this time was that when the virus infects, attaches, and then penetrates and injects the viral DNA in there, there are going to be some enzymes that actually break apart the bacterial chromosome. And so you can see that there now it's in pieces. They colored them in blue so that we can recognize there's the DNA from the bacteria, and the red is the viral DNA. Now, when the bacterial machinery now is, metabolic machinery is now synthesizing new nucleic acids, viral nucleic acids, the viral DNA, the proteins responsible for making the capsid and so on, synthesizing all the parts that are needed to put together the virus. The virus is now going to be assembled. That's one of the last stages of the lytic cycle before the virions are released. Well, it just so happens that because you have the original bacterial chromosome that's been broken up into pieces, some of the capsids are going to accidentally pick up fragments of the DNA from the bacteria instead of picking up the viral DNA. And so in this image here, it's kind of small here, the virion that's being released on the left has a red colored DNA in there to represent the viral DNA. And then the blue one here we have one that's accidentally picked up the bacterial chromosome, or piece of the bacterial chromosome, piece of bacterial DNA. Now the capsids are the same. So the capsids are going to interact with other bacteria when they're released in the exact same way. They're going to attach, and then they're going to inject the DNA during the penetration stage, that DNA into the host. The problem here is that when this virus now, virion, that's now carrying a fragment of the host bacteria's DNA goes and attaches, now we're injecting not a viral DNA, you're injecting a piece of the bacterial DNA from the prior bacterium. And that once that enters, now we're going to have a similar process that occurs during transformation that we saw earlier. That segment now is going to act like a plasmid. It's going to go in there and integrate into the host cell's chromosome. And now we're not going to even be making viruses, but this bacteria now may be now different because that piece of DNA be carrying different genes on there, or different versions of those genes, and so now this bacterium is now going to be phenotypically different. Okay. Now, an important thing to remember here is that virtually any one of those fragments of DNA can accidentally get taken in into the capsid before release, and so here just about any gene can be transferred, and that's why it's called generalized transduction. Okay. If we go to specialized transduction, we need to recall a bit about the lysogenic cycle. And in the lysogenic cycle, remember that we're still going to have attachment that occurs, okay? And then you're going to have the penetration occur there. But instead of going into the lytic cycle where we start making new viruses right away, instead, this viral DNA is going to become integrated into the host DNA. So let's say here's your normal host bacterial cells DNA, and then your viral DNA gets put in there. And I'm just going to draw a zigzaggy line to represent the viral DNA. So this would be your viral DNA, and remember that's going to be called the prophage or provirus, and this is the rest of the bacterial chromosome, and this is within the cell, the entire bacteria cell, right? That entire bacteria cell is called the lysogen or lysogenic bacteria, right? So now every time this bacteria divides, you're just reviewing the lysogenic cycle, we got to make a copy of that chromosome, and when you're doing that, you're making a copy of the prophage. At some point, though, induction occurs, and the prophage excises out. The problem here is that during this excision, it's imprecise, and you end up getting a piece of the chromosome from the bacteria. And so when this occurs, then you start making copies of your virus, and the virus is now carrying a piece of the bacteria's chromosome. Now, during this part here, it is not any gene that can be transferred. It's usually only the prophage usually inserts itself in very specific location within the chromosome. So usually it can only be certain types of genes that get transferred here. So it's not any gene here. It's specialized transduction. And it's usually some of the genes that are located right near the place where integration had occurred. So it might be just the genes that are right nearby there. So that's the difference between the two. One involves lytic, that's generalized transduction, and any gene can be transferred. For lysogenic, it's not any gene, it's only certain kinds of genes that are near the place where the integration had occurred. Then you have conjugation, and conjugation occurs when one bacteria, which we're going to call the F+ bacteria, is carrying a plasmid, and that plasmid has instructions for building a conjugation pilus. So let's say this is the F chromosome, and it's carrying a little plasmid, a little piece of circular DNA, and then here's your normal bacteria circular chromosome all coiled up because it's huge. And this one is an F+ cell, and there are instructions now to build a conjugation pilus. And here's another bacterium, and it has its own chromosome, but it doesn't have the plasmid, and this is going to be referred to as the F-. The F stands for fertility factor. Okay? Now this process is going to occur, and when the plasmid is being transferred, it's not sending the entire piece of DNA, it's only sending one of the strands. Remember, DNA is double-stranded here in this case. And so one of the strands is going to get transferred, and it's going to begin by the production of a conjugation pilus. And on the plasmid, there's going to be an origin, okay, a certain section of it. So I'm just going to kind of try to highlight. So there's your origin right there. That origin attaches to the cell wall and then begins to transfer only one of the strands. So you can see that process is progressing in the next picture there. And one of the strands of the DNA from the plasmid is going to be transferred into the F- cell. While that's occurring in the F+ cell, or the original carrier of the plasmid, you're going to have DNA polymerases going in, remaking the strand that's being transferred. And what it's going to do is going to use the strand that remains as the template for making the new strand. When that the strand that's being transferred gets into the F- cell, DNA polymerase is going to begin. You can see they're coloring the new strand purple here. Is going to begin making the other strand in the F minus cell. The end result here of this, what's called rolling circle replication, is you're going to have two exact copies of the plasmid. Both are now going to be F plus cells and are capable of transferring their plasmid to another F minus, to other F minus cells. And you can see that both plasmids are made of one old strand and one new strand. They color-coded the new strand in this case to be the purple strand, and the orange strand is the old strand. So that's the case there. Now, once that plasmid is in the cell, these F plus cells can go and conjugate with other F minus cells and transfer the plasmid, or that plasmid can integrate, just the way a viral DNA integrates into the bacterial chromosome for bacteriophage here. The plasmid can integrate in there, so the process is called integration, same name. And here you're going to have a special segment on the plasmid and on the bacterial chromosome called the insertion sequence, a certain sequence of bases on there, and they recognize each other. So the plasmid goes there, and then you're going to have the integration occur there. And so the integration occurs there, and now the entire, so they're not showing the entire bacterial chromosome, it's a lot larger, it's a big loop, right? And so here now it's integrated in there, and the rest of the bacterial chromosomes in purple. And they're now going to call this cell, it's not an F plus cell anymore, they're going to call it an HFR, which stands for high frequency of recombination here. And so this cell now can go on and do the things that give it the name high frequency recombination. I'll tell you what that is right now, but let me say that before we go into that, let's talk about the fact that the plasmid can be cut out again, and that process is called excision. Okay, we also saw that when it came to the replication of virus in the lysogenic cycle. The viral DNA can be excised out. This is not viral DNA, this is just a plasmid, but the plasmid can be excised out using similar enzymes to get it out. And so we can cut it out, and once we cut it out, it's just the opposite of integration or a reverse process. And then there's your free plasmid. Remember, this is inside the bacterial cell, and we now refer to the cell as an F plus cell again. Okay. So what is the difference then between an F plus cell and an HFR cell, or high frequency recombination cell? The difference here is that an F plus cell will only transfer the plasmid. Okay? The instructions are still there to transfer the plasmid, but this plasmid is now part of a larger chromosome. So an HFR cell cannot just transfer the plasmid, it can also transfer its entire chromosome if it's allowed to. And that's where you're going to get recombination occurring, because if it transfers any part of its bacterial chromosome into an F minus cell, it's sending pieces of its own DNA in which the genes that are on there may contain different alleles. And those different alleles go in there and genetically recombine in F minus cells. So that's where it gets its name from. Now, going back to the F minus cell, I mean, the excision process. Here they're showing a perfect excision. Occasionally, the excision may accidentally take a piece of the original chromosome out. So this is a perfect excision, but imagine a case where the plasmid takes an actual piece of the bacterial chromosome in there. And in that case now, this new plasmid is not the original plasmid, and we're going to refer to it as an F prime plasmid. And the F prime plasmid is now contains a piece of the chromosome beyond just what the original plasmid was. Now, when we look at the HFR ones here, you're still going to have that origin in which the transfer is going to occur. It was there on the plasmid. It's still there on the plasmid that's now integrated there in the HFR cell. So this HFR cell can still form the conjugation, can still go and conjugate with other cells. So here now, the HFR cell can still form that conjugation pilus with an F- cell, but instead of just transferring the plasmid, it can transfer its entire chromosome if allowed enough time. Now, the transfer takes time, and it's still going to be rolling circle replication that it would occur. You're only going to transfer one strand from the DNA. But the bacterial chromosome is large. It's so large that I think for transferring the entire genome of E. coli in this example, the E. coli, it would take about 100 minutes for the entire chromosome to be transferred. It would take much less time for just a small plasmid. And this brings up this next part here, is that when conjugation occurs with an HFR cell, you can interrupt the process at different points, at different times. And you can actually determine which genes, the order of the genes, because during the transfer, during conjugation, you're always going to go in the same order. It's always going to be at the same origin of replication, and there's a polarity to the molecule. It's always transferring, always in one direction along that circular chromosome. So as that chromosome is being transferred, if you interrupt this process during that transformation at different times, and then you analyze, well, what was transferred, you can actually determine the order of the genes on the chromosome, and that gives you a map of the genes on the chromosome. So this was the part on how can you map genes. Well, first of all, you need to be able to recognize the genes. So they have an HFR, which have that plasmid within the bacterial chromosome. And then they have F- bacteria as well, and they mix them together. And so the HFR is going to form the conjugation pilus, and they're going to start to transfer their entire chromosome now into the F- cells. And we have to be able to recognize that. So they just have an example here of 4 different genes, and they have them symbolized differently. Here you have a lac minus, and here's a lac plus. So these are 2 different alleles for the lac gene. It's a specific gene called the lac operon gene. And there's 2 different versions, so you can actually track. Now you know that your gene or your allele has been transferred during conjugation if you see it appear in the F- cells as they now are picking up the pieces of the chromosome. So let's say you get a whole bunch of test tubes and you start them all at the same time, and you're mixing HFR with the F- together, so they all start conjugation at the same time. And then at different times, you're going to put the contents of that test tube in a blender to agitate it, and that breaks the conjugation tube. And then you go and you analyze the bacteria from there, and you determine, well, what genes do they have? And you can see, if you do this at different times, you can see that the different genes appear at different times. The AZI gene is going to appear at about 8 minutes into conjugation. Then you have the TON gene at 10 minutes. That means it's further along. And then the next gene that, of the ones they were studying appears, the lac operon gene, at about 15 minutes. And then the GAL gene at about 17 or 18 minutes. And so here now we have a map of the location in minutes. So the units are minutes. So how many minutes along the chromosome? So that's how they map the chromosomes. It's pretty interesting. And again, this uses the high-frequency recombinant bacteria cells, and they have, remember, they have that plasmid within their chromosome. And then there's antibiotic resistance here. Antibiotic resistance can occur when a bacteria picks up a plasmid that has resistance genes on it, or genes that provide resistance to the bacteria that's picked it up. An example here is Staphylococcus aureus, which is a normal bacteria found on your skin. It can cause skin infections. It's a normal part of your flora there. But if you have a break in your skin, it gets in there, it can cause an infection in there. A skin infection, it can get worse if it gets into your bloodstream and you get a systemic infection overall. But there are some basic antibiotics that can help clear out that infection. But there are some resistant strains that have become very dangerous overall. And so in cases where plasmids provide resistance, they're called R plasmids. Now, some genes from pathogenic species can be transferred either as plasmids or as transductions. So let's say you have a pathogenic species and there's a gene on there that produces some toxin that creates a virulent situation, or a situation that could create disease when that bacteria gets into some host overall. And so those genes can be transferred by plasmids or by transduction. So here you can have a virus, bacteriophage, that ends up transferring that by transduction. And this is the case for Enterobacterium in the Enterobacteriaceae. There's a strain called E. coli O157H7. And this one is acquired genes that give it pathogenic traits. So E. coli is normally not pathogenic. It's normally part of our flora, but then ends up picking up a gene that makes it virulent and very dangerous. So mutations can arise in bacteria spontaneously, or they can happen at much greater rates if there are things in the environment that promote mutations, like radiation or certain kinds of chemicals that can cause mutations. The mutations are the way in which new alleles are produced to begin with. But if you have things in the environment, they can increase that. There are some bacteria that are referred to as auxotrophs, and for these bacteria, the auxotrophs, they're nutritional mutants. And here, these bacteria have ended up getting a mutation in which they're no longer able to produce certain nutrients. Let's take amino acids, for example. Let's say that you have bacteria that are capable of synthesizing all 20 different amino acids that are used to make proteins, and then they have a mutation for one of them. If that's the case, then they cannot just be grown on they can't just grow in an environment that's poor in nutrients. They can't just be grown with sugar. You're going to need to have those nutrients in which they can no longer synthesize anymore. So they can't just survive on a basic energy nutrient medium. And then the mutations themselves can spread in a bacteria population rapidly, because bacteria divide very easily. And so the mutation may be transferred by a plasmid, for example. And this is the case has been observed with the Staphylococcus aureus. Again, that's normally found on our skin. There's methicillin-resistant Staphylococcus aureus, and then a very powerful antibiotic, vancomycin, as well. And this is pretty bad, because this normally harmless bacteria, again, it can create infection. It gets inside, and then if it spreads within your body, that's really bad if there's no antibiotics that can help fight off the infection. Then there's the CRISPR system that has been observed. Bacteria have likely been influenced by viruses forever. I mean, bacteria have been around for quite some time, going back 3.5 billion years. So it makes sense that over time, bacteria have developed ways to help fight off viruses that can attack them. And so one of those ways is a more recent understanding, in that the prokaryotes, the bacteria, have within them, within their genomes, they have these repeating spacer regions that are referred to as CRISPRs. And that's an acronym short for clustered regularly interspaced short palindromic repeats. And that's within these regions where within this region where the bacteria is going to store short segments of viral nucleic acid. So when they've been infected by a virus, they do have enzymes that can go and break apart the viral nucleic acid, and in some cases they'll end up storing a segment of that viral nucleic acid, and they'll store them within those regions. A locus is a region within a chromosome, so they'll store them between those CRISPR regions, and that segment of viral nucleic acid can be used to produce RNA. That RNA would have that specific sequence that recognizes the viral nucleic acid because of that sequence. And so that could help locate any new viral DNA that's gotten into the bacteria, and these short segments are going to go in to help degrade or break down that viral nucleic acid. So this section is going to look at metabolic diversity in prokaryotes. And the first objective is to describe metabolic strategies that are used by prokaryotes to get carbon, energy, and sources of energetic electrons, which are reductants. And then to compare and contrast the ways prokaryotes can obtain their energy, electrons, and reduced carbon. Reduced forms of carbon are your organic molecules, things like carbohydrates, proteins, things like that. So we want to look at nutritional strategies to start with. And the nutritional strategy for an organism is the way it acquires its carbon, the way it obtains or gets its electrons for redox reactions, which are reactions that either gain or lose electrons, and also what the source of the energy is to drive these processes. So for obtaining energy, you're either a chemotroph or you're a phototroph. A phototroph is going to harvest light. It's going to use energy from light to help drive these processes. For chemotrophs, they're basically going to be oxidizing reduced chemicals. Reduced chemicals are going to be ones that have energetic electrons, and when you oxidize them, you're taking the electrons away from them, and then you're going to use those energetic electrons. So in general, chemotrophs are going to oxidize compounds to get their electrons. Then there's two kinds of chemotrophs. There's lithotrophs and organotrophs. Both of these are just subcategories of chemotrophs. For lithotrophs, the electron source is inorganic. Okay. For organotrophs, the source of those energetic electrons are going to be from reduced forms of carbon, organic molecules, pretty much. So what are the sources of carbon? And carbon is going to ultimately be needed to build biomolecules. And if you're an autotroph, the source of carbon is inorganic carbon dioxide. So you can have, using some of the terms from up above here, photo is going to use light. Litho is inorganic. And so this is going to use energy from the sun, the photolitho autotrophs. We saw these with, for example, plants when you were studying photosynthesis. The inorganic source of those electrons is water. Carbon dioxide is used as an inorganic source of carbon. Now, in order to get those electrons, those sunlight energy is required to help split water molecules to gain those electrons. And then chemolithoautotroph, here again, you're going to oxidize inorganic substances, and your source of carbon is carbon dioxide. For heterotrophs, generally that meaning hasn't changed, even though we're using bigger terms now. The source is organic molecules for your carbon. Now, this kind of metabolism right here is unique to prokaryotes. It's called photoorganoheterotrophs here. Light is an energy source, but they're going to be obtaining their carbon and electrons from organic compounds. So that's an odd sort of nutritional strategy. And then we have chemoorganoheterotrophs, which more adequately describes what animals are, and animals include humans as well. So here, both your carbon and your energy comes from your organic molecules like carbohydrates. So when we look at prokaryotes, we want to look at the way they respire, their cellular respiration. And cellular respiration is the way a cell goes about obtaining and acquiring and transferring its energy for use in the cell. And a lot of times that involves the transferring, or many times we have to be transferring energetic electrons from one place to another, ultimately to generate ATP. And so a helpful little quick review looking at eukaryotes, which is not really the subject of this chapter, but you did study respiration in eukaryotes. So I pulled a quick picture of a eukaryotic cell with its mitochondria in there. And the mitochondria is involved in aerobic respiration. So for eukaryotes, the key here is that when it comes to respiration and how they take the energy from these organic molecules, they're gonna need to use organic molecules as electron donors. The way they take that and then transfer that energy to ATP, that's your cellular respiration process, requires organic molecules, and you're going to need oxygen as your final electron acceptor. So that's the key here. So eukaryote is aerobic respiration. You can start with glycolysis, and glycolysis is going to make 2 pyruvates here. You're going to also get 2 ATPs out of it. And then the pyruvates go in there and then go through Krebs cycle. And then what's happening during this time is you're oxidizing the food, the organic molecule, and you're getting these energetic electrons. Those energetic electrons are being transferred to an electron carrier called NAD, and there's another one called FAD. Let's just look at NAD to make the discussion simpler. This is the oxidized form, and if we add 2 electrons, anytime you add electrons, you're reducing, right? So you add 2 electrons and you add 1 hydrogen ion, or a proton they call them, and you end up with a reduced form NADH. This is your electron carrier, and it's carrying those high energy electrons, and the NADH is produced both in glycolysis, which doesn't require any oxygen. It's also produced during the Krebs cycle as well. So there you can see your NADH is being produced. And those NADHs are going to deliver their 2 electrons to the electron transport chain. And that electron transport system is located on the inner membrane of the mitochondria. Remember that mitochondria has an outer membrane, inner membrane. And those electrons are used as the electrons are moving down the transport system. That energy and the electrons used to pump protons in here. And that generates a great deal of potential energy, which can be used to generate a whole bunch more ATP. You generate only a little bit during glycolysis, but a lot more if you go to electron transport, but you can only do that if you have your oxygen as an electron acceptor. Okay? So oxygen accepts those electrons along with some hydrogen ions, and you're going to get 2 hydrogen ions and 1 oxygen, and that gives you water as a product from that. That's eukaryotes. For prokaryotes, they're more diverse in where they can get their electrons from. So your electron donors can come from inorganic materials or inorganic substances. And then your final electron acceptor can vary as well. So your final electron acceptor could be, it's not going to be oxygen, so this would be anaerobic respiration. So final electron acceptors can include nitrate, sulfate, and carbon dioxide. A nitrate is a compound NO3 with a negative charge. Sulfate is SO4 with a negative 2 charge, and then carbon dioxide. And so I found this diagram that comes from industrial research. I was just looking for something real quick to show. And in these spherical bubbles represent certain types of bacteria. This is acidogenic bacteria. That means it generates acetic acid. Here's acidotrophic, which uses the acetate ion as a source of electrons. Here you have your hydrogenotrophic methanogens. And here's a great example, is that the electron donor is molecular hydrogen. So molecular hydrogen has electrons, and your electron acceptor is going to be carbon dioxide. So the electrons get transferred from your hydrogen to your carbon dioxide, and that's going to end up producing methane as a product. So that's why it's called a methanogen. And so here's an example of anaerobic respiration in which your electron acceptor is not oxygen, and your electron donor is not some organic molecule. Over here we have a sulfate as our final acceptor here. Again, hydrogen is your donor, and there's your sulfate accepting the electrons. And then your products would be something like hydrogen sulfate. In this case, this comes from industry, and it shows how these bacteria lead towards corrosion of metal pipes. And then we have fermentation. And so for fermentation, this is incorrect right here. We should say lactic acid. For fermentation here, in the case where you're using organic compounds as your electron donor, source and bacteria can do this, by the way. So some bacteria do use organic compounds, right? So they're chemoorganoheterotrophs. And so they're going to go through glycolysis, for example, if they're going to break down sugars. And glycolysis is always going to give you pyruvate. It requires no oxygen. And the thing is, the glycolysis requires that electron acceptor, that NAD+, or that electron carrier. And so if in this case here, for these bacteria and these and eukaryotes that go through this aerobic respiration, if they run out of oxygen, then what are they going to do? Because what they're going to end up doing, they're going to keep going through glycolysis, doesn't require any oxygen, and they're going to keep generating 2 ATPs only, and then they're going to be producing NADH. And NADH cannot go into the mitochondria unless you have oxygen as your final acceptor. Okay? So we can't go through here. We can't get all these ATPs. We can get a little bit of ATP, but eventually you're going to run out of NADH because the electrons get tied up on that molecule. They're supposed to go get delivered to electron transport. So if you run out of NADH, then you can't even do glycolysis, and you make no more ATP at all. And then all metabolism stops. Okay, so what is the answer there? Go through fermentation. And so for fermentation is going to use an organic molecule as an electron acceptor. So in other words, the organic molecule is going to take the electrons off of the NADH so that we can free up the NAD to continue to run glycolysis. So for eukaryotes, they're pretty much limited in what those acceptors are and what they end up producing. So for eukaryotes, they produce lactic acid or ethanol. Ethanol is the alcohol you find in wines and beer. Lactic acid, our muscles are capable of doing this, but that's about it. No other types of tissues can really do this to any real extent. But other microbes can produce lactic acid, yeast, which are single-celled fungi. For prokaryotes, though, they're going to generate pyruvate when they're breaking down organic sugars, but when they ferment the pyruvate, they're going to produce a lot of other kinds of acids and alcohols, and not just lactic acid and ethanol. So they're not limited in what their products would be. So they can produce acetic acid, they can produce methanol, propanol, different kinds of products. And some of those products from these bacteria are used industrially and commercially to make things. A lot of food products are bacteria are used to help flavor the foods and so on. This is an image just showing the difference between lactic acid fermentation and alcohol fermentation. An extra step is required for alcohol fermentation. We have to remove a carbon from the pyruvate. It's normally a 3-carbon sugar compound, the product of glycolysis. CO2 is released, and that leaves a 2-carbon compound called acetaldehyde, and then the electrons are added on there from the NADH so that we can regenerate NAD. When we put the electrons and the hydrogen on there, you get ethanol. So we're going to focus a little bit now on microbial ecology. And ecology is the study of the relationships of organisms with their environment. So your first objective is going to be to describe the role of prokaryotes in biogeochemical cycling of elements. The cycles that take place in cycle carbon and nitrogen and so on. And then we're going to explain how the tissues of animals and plants can be considered microbial ecosystems. And then describe the role of prokaryotes in what's called bioremediation. Bioremediation is using these living organisms like bacteria to help fix problems that occur in the environment. First things, looking at the first objective, prokaryotes do play critical roles in cycling materials in the environment, in the ecosystem. They're decomposers, and that means they're going to break down dead organisms, and that's going to release carbon and phosphorus and so on back to the soil or back to the atmosphere. When they're breaking down, they release CO2 into the air. Bacteria are also important at fixing carbon and nitrogen. And when you fix carbon, you're taking carbon dioxide in and you're reducing it. You're making larger molecules out of it. And photosynthesizers do this. They fix carbons into sugars. And cyanobacteria do this as well. And this process can add oxygen to the atmosphere. And then the nitrogen fixtures are going to take what's pretty much an inactive form of nitrogen, which is atmospheric molecular nitrogen. It's fairly stable. And certain bacteria have an enzyme that allows you to fix that nitrogen and end up reducing it to ammonia, which is more useful to plants. In aquatic environments, there's a cyanobacterium called Anabaena. And Anabaena is a genus, and they have their cells are capable of fixing the nitrogen. And in the soil, there's one called Rhizobium, which is associated with roots. We found a couple of biogeochemical cycles here, and here you can see carbon cycling in both in the water and in the soil and in the oceans. You can see carbon taken in, carbon released, and so on. And overall here you can see microbes are playing a role in their microbial respiration. It's going to release CO2. Microbes are also going to break down or decompose. And this shows more of the carbon cycle that involves more than just these microbes. There's also these photosynthetic bacteria called cyanobacteria that can take in carbon dioxide and fix it as well. So both decomposition and fixing. And then you have the nitrogen fixation, and so you'll have your nitrogen fixing soil bacteria right here that'll take atmospheric nitrogen and then use it to form ammonia and the reduced form ammonium. And then that can be taken in by plants to build things. Other bacteria can change that or convert that to nitrites and nitrates, which can also be used by our plants, taken in and then used to build important molecules like proteins and nucleic acids. Plants themselves and plant tissues can harbor microbes in there and themselves become these ecosystems for bacteria. There on the right you see roots that have these little nodules that are produced when bacteria invade the tissue of the root tissues there. And this is a mutually beneficial relationship. Bacteria in the genus Rhizobium end up creating these nodules there on the roots. And then what happens here is that the Rhizobium fixes nitrogen, which is like a fertilizer. So the plant benefits from there. And then the plant gives energy nutrients to the bacteria. So it provides a food source for the bacteria. So that's a mutualism. In other cases, we have parasitic relationships. That describes another ecological relationship in which some bacteria, I think one is in the genus Agrobacterium, ends up producing these galls, which are tumor-like structures. And basically the bacteria promotes the growth of excess tissue. And that can block the flow of materials within plants, and that can reduce growth of the plant and even kill the plant. So this one is called crown gall disease. You can see that gall or tumor-like structure. And then animals are also microbial ecosystems. There's ruminant mammals like cows and goats. They have multiple stomachs in there, and they have these microbes that live within their guts, and they help the animal digest food and get nutrients from the food. And those microbes have a nice place to live with plenty of food for themselves as well. We also have important bacteria that live in our digestive systems and even on our bodies. And overall, this nice healthy balance helps reduce the growth of harmful bacteria and even fungi, and even help promote digestion and even provide vitamins to us. There's some bacteria that help produce vitamin K in your large intestine. And then there's bacteria that are associated with animals, including humans, that are parasitic, and many times in harming the animal by taking nutrients from it also can cause disease. So not just parasitic, but pathogenic. And then when things go wrong in the environment, whether it's human activity or natural activities that can create problems in the environment, pollution, we might use bacteria to do bioremediation. Remediation is just a fancy way of saying let's fix the problem here. So they can use bacteria for bioremediation. These bacteria can remove pollutants from the water, air, and soil. For example, when you flush your toilet, the waste goes to sewage treatment facilities where they use bacteria to help decompose the solid waste, converting it into less harmful materials, totally recycling everything back. When we have large oil spills, which are ecologically a disaster, you can see just with this image of this bird completely coated in oil is likely to die along with anything else that's there. So that those oil spills, large amounts of oil in the area, can be an ecological disaster. And it turns out that there's bacteria that have evolved that are capable of breaking down these large hydrocarbon type molecules because of plants that produce similar types of compounds over eons of evolutionary time. And so we can figure out a way to work with these bacteria to sort of help clean up. Now, it's always better, prevention is always better, any of these types of disasters. They give an example of the Exxon Valdez oil spill in Alaska, and you can see them here. They're spraying down these rocks here, trying to remove a lot of excess oil. But the thing here is that they want to provide some nutrients like some phosphorus and some other things that would help these bacteria grow. And then they can help break down the oils and these heavy hydrocarbons that are there. There's also other bacteria that help remove halogenated compounds. Halogens are found in the second to the last column. They include chlorine and fluorine. And a lot of these compounds are pretty toxic, created in industries. But there are bacteria that are capable of breaking down these compounds into less toxic materials. And when they're doing that, they're actually using the halogens within their metabolism as electron acceptors, and so this is called a halorespiration, and so that can be useful. This section looks at diseases in humans caused by bacteria, and your first objective is to describe how bacterial pathogens cause disease, and then to explain how Mycobacterium, tuberculosis, and Helicobacter pylori, which are two types of bacteria, can cause disease, and then identify sexually transmitted diseases caused by bacteria. These should actually be underlined or italicized since they are scientific names, the binomials. So in the early 20th century, so we're talking about the early 1900s, bacterial diseases, infections did kill like 20% of children before the age of 5. So that was pretty terrible. Part of the reason was there was not very good sanitation to get rid of our waste, and that created a lot of contaminated drinking water situations. And also, antibiotics were not really a thing then. But in recent years, now that this thing is improved now because of better sanitation and antibiotics is, I guess, the point. But in recent years now, many bacterial diseases are starting to appear or reappear that haven't been before. And one of the reasons is that many of the diseases that were fought off with antibiotics, these bacteria are now becoming resistant to these antibiotics is one of the reasons. So here's a table of some important human bacterial diseases. The first column is the disease, the name of the pathogen, the bacteria, your vector, or where you're likely to find that type of bacteria, and then the epidemiology for it. Anthrax, as mentioned earlier, is caused by a pathogen called Bacillus anthracis, and the vector reservoir are animals, including even skins produced by animals. Here, the bacterial infection can be transmitted through contact or ingestion, and it can be fatal. Then there's botulism. The toxin produced by Clostridium botulinum can happen in improperly prepared food, where the spores of this bacteria, we mentioned them earlier, the spores of them can get trapped in food, and then without oxygen, they start their anaerobic metabolism and then produce these compounds that cause paralysis, and this one can be fatal as well. Then you have sexually transmitted disease, chlamydia. It is a STI, or sexually transmitted infection, and it's becoming more increasingly common in the last 30 years. Then we mentioned cholera in the virus chapter. The Vibrio cholerae is the bacteria and seems to go through this conversion whenever the bacteriophage goes into a lysogenic cycle with this bacteria. This can cause severe diarrhea and very high death rates, 50% mortality. And a main problem here is contaminated drinking water. Dental caries or cavities, and those are caused by bacteria, Streptococcus type, two different kinds of species. Then there's diphtheria, caused by a bacterium called Corynebacterium. Diphtheria, in humans we get vaccines for this one, along with the tetanus vaccine, the DPT shot. This one causes acute infection, inflammation of the respiratory mucous membranes, and it can be easily transmitted. Gonorrhea is another STI. here, and this is increasing worldwide. It's usually not fatal. And then Hansen's disease or leprosy. This is caused by Mycobacterium leprae, as in humans and in wild armadillos. This is a chronic infection of the skin. And then there's Lyme disease. It's caused by a pathogen called Barilla burgdorferi, and this is spread by ticks, and deer and rodents are reservoirs for it. So the tick is the vector, where the tick drops off of one host and then goes on to another one and then transmits the disease that way. You get lesions, malaise, fever, fatigue, stiff neck, and headache from the disease. Then you have peptic ulcers caused by Helicobacter pylori, and antibiotics are used to treat that one. Plague, which you probably heard of, a study of history perhaps, caused by Yersinia pestis. Fleas are the vectors, and the reservoirs are rodents, rats, and squirrels. Squirrels are actually rodents as well. This caused the plague in Europe in the 14th century, and this is endemic in wild rodents in the western United States, so we find plague here. And then there's pneumonia, and that's can be caused by viruses, but can also be caused by several different kinds of bacteria. This is acute infection of the lungs, and some vaccines are available. Tuberculosis is one in which antibiotics are becoming resistant to its Mycobacterium tuberculosis. And there's acute bacterial infection of the lungs, lymph nodes, and meninges. The meninges surround the brain and the spinal cord, and this incidence has been on the rise, but is now falling. The infections are complicated by drug-resistant bacteria. Then there's typhoid fever caused by Salmonella typhi, and then typhus caused by Rickettsia typhi. And their epidemiologies are there. Just scan over this. Don't go memorizing every disease that's on this table. This is a short list to begin with. We're going to focus on some other ones here. So, in answering one of the objectives, how do bacteria cause disease? If they cause disease, they're pathogenic, and one of the first things, they have to gain entry to the body. So they got to get into the body in some way, whether that's sticking to mucous membranes or entering a wound. Then they're going to colonize at the infection site, and they have to evade the immune system. They can get by the immune system, they have room to grow. And then they can spread to other sites in the body. And the way they can cause damage is by producing toxins or triggering an inflammatory response. The toxins themselves can spread out and cause problems, like the toxin that causes botulism, cause paralysis. That toxin spreads throughout the body and causes paralysis. Inflammation can also be a problem because that can damage tissues as your body is trying to fight off. The inflammation is actually damaging some of your own tissues in an effort to fight off the infection. You gotta damage a few things to try to get better. These bacteria do have structures or physiological processes so that they can use the host as a niche to survive in there, and then they can end up causing disease. Tuberculosis, or TB for short, is called the scourge for 1,000 years. Again, it's caused by a bacteria called Mycobacterium tuberculosis. It affects the respiratory system, and it's easily transferred from person to person through the air when you cough, and the bacteria gets aerosoled into the air. And the problem with this one is that it's developed a multi-drug resistant strain. So it's very, very difficult to treat because the bacteria doesn't get affected by very many different drug types. Then there's the H. pylori, short for Helicobacter pylori. This causes stomach ulcers. And this can be treated by reducing stomach acid, also treated with antibiotics. The pathogen can also cause stomach cancer and a type of lymphoma. So long-term, this can get pretty bad. And then you have your STIs, sexually transmitted infections, and then if the infection leads to problems, then the disease occurs there. And so gonorrhea is one of those. This is one of the most prevalent communicable diseases here in North America, caused by a bacterium called Neisseria gonorrhea. and it's transmitted through exchange of body fluids, and it can pass from mom to baby during childbirth. And then there's chlamydia, caused by Chlamydia trachomatis. It's called the silent STD, and incidents have skyrocketed here, and silent because don't always know you have it until perhaps later. This can cause pelvic inflammatory disease in females and result in damaging the heart through certain mechanisms, disease mechanisms. And then there's syphilis. Scientific name of the bacteria that causes syphilis is Treponema pallidum. And this is transmitted through sex or contact with open cankers, these sores that are produced by the bacterium. This can also pass from mom to baby via or during childbirth. There's 4 distinct stages. The first 2 are highly infectious. There's the canker stage. This is the first one where the little sores are produced. And then the second stage is the rash, also infectious. And then there's latencies. This can go on for a while. Doesn't seem to be any issues going on. You're no longer infectious, cannot transmit it. But at this point here, the bacteria is attacking internal organs. And then in stage 4, the damage is now evident with heart disease, problems with your nervous system, and mental deficiencies because of the infection. This is a graph just showing the incidence in epidemiological numbers here. A lot of times they'll report epidemiological problems like with a virus or even auto accidents have their own epidemiology. And they usually report them, the numbers per 100,000 people, for example. So these numbers here on the scale are the numbers per 100,000. So this would be 50 per 100,000 individuals, all the way up to 500 per 100,000 individuals. And so you can see that gonorrhea has been on the decline from 1984 to 2014, that the data is available for this graph. And syphilis has remained relatively stable there in the number of cases, the number of new cases reported. The one that's been on the rise has been chlamydia. And that one has been growing steadily from 1984 all the way to 2014.